Welcome to Chapter 9. This is mini lecture number 3. So we've left off and we're going to now look at hybridization and specifically, to corn. But the first thing that we kind of need to understand is what a hybrid is. So a hybrid is just the offspring that results from a hybridization process. So it's you really hear that when you're buying different plant or different seeds to grow in your field. So when we're looking at hybridization, we have to look at how that corn inbreds by either self-pollinating and selected by the corn plants for several generations. So from that F1 to that F6 generation to make a very uniform variety. So F1 looks way different than F6 because F6 is going to be very uniform. We're going to have almost the same growth. We're going to have the same development. It's going to be they're all going to be the same level. They will all have the same really growth aspects to them. So we have to do that by crossing two inbred plants with that very distinct genetic trait. So the first hybrid, again, that F1 is typically has those traits that are superior to those that have either been to the inbred parent. Okay, so we're taking that F1 and trying to get it to that F6 that we can then take the seeds and then. So this is just an example of a hybrid corn production. So you can see on the top there, so as time goes by, on the top we're decreasing that vigor and increasing that homozygosity, and then it's inbred into that hybridization. So we don't want that. We want it to be uniform. Okay, that's obviously not uniform by time. So when you're looking at the bottom, you can see the inbred A versus the inbred B, and then we're seeing that hybrid corn plant, and that's what we're going to grow. That's what we will be known as a hybrid that you would buy it for seed. So genetic transformation, how do we use it? Why is it important? The really two main things that we look at is how that gene originated from all of these many different sources of different plants, and then transferring then occurs from either a bacterium or a gene gun, which I'll show you a pretty cool graphic about. It's also in your book as well, and we'll look at that. And then we use that for the qualitative traits like a herbicide or a disease resistance. Unfortunately, in this section of the United States, we have to look at a lot of those because of different herbicides that we see, different diseases that we're starting to see pop up, and now different pests that we're starting to see pop up. So it's really important to look at that. So when you're talking about the difference between a gene transfer and a bacterium and a gene gun, we'll look at how we use both of those. Okay, so agrobacterium, or the bacterium, we're looking at the gene that's identified by that's identified and isolated. So same thing with the gene gun. So it's isolated, it's not going to be able to really go anywhere. In the bacterium, it's just going to be inserted into that cell, into that plasmid, and nothing's really coating it, nothing is really messing with it. It's just there, that specific gene. Okay, so then you go through that genetic process, and it will then be, those cells will be screened, and that new gene will be then selected by those transforming cells because it has a marker. Then it will be a single cell transformed, and then that transgenic plant will then regenerate from that cell. It's kind of the same thing in the the gene gun, but the gold particles are put within that, they're coated within that DNA, and then the cells are then shot with that gene gun that we talked about and you've read about, and the DNA is then incorporated into the plant cell chromosome. Okay, so it's shot right into it. So we're taking out the really mixed part of the plant cell. We're taking out a little bit more of that time. We're taking out a little bit more of what that process has to go through to then get to that transgenic plant. So stack traits, why are they important, and why is it important to know about them? So there's a crop with more than one transgenic trait we call a stacked trait. Okay, so we're stacking it up really. And that's really important because we look at herbicide resistance in comparison to insect resistance. So if I'm going to buy a variety of a soybean, I want to make sure that it has a herbicide resistance and an insect resistance. So I'm stacking that trait. A corn plant, okay, can be stacked with 3 traits, and sometimes with 4 traits, or a quad stacked. So, but that's a little bit too much money. It's a little bit expensive. So we personally don't buy those quad stack traits, but a lot of other people do, which is awesome. And so you can actually have 8 or more stacked traits, and so that's getting those traits pretty up there. So what's the difference between genetically modified organisms, friend or foe? So what's the difference between the benefits and the concerns? Now, when we look at this in class, we're going to really decipher what our opinions, our educated opinions will be, and then we'll look at how we have to then be very cautious of other people's opinions, and we have to respect them. So I'll tell you this in class, that I'm going to shake your hand and agree to disagree. If we don't agree, I'm going to love you the same. I'm going to like you the same. But even if your opinion is not the same as mine, I'm going to shake your hand and I'm going to agree to disagree, because I believe it's so important with different aspects of agriculture like this that can kind of separate us, which is not good, because agriculture is kind of hit on a lot in terms of funding, in terms of marketing, and how we're looked at within the broad spectrum of agriculture. So it's really important to stay together. So again, shaking your hand, agreeing to disagree if you don't have the same opinion as someone else. So what are some benefits? Just some 3 big ones in my book. So crop resistance to pests. I have unfortunately seen a lot more pest damage than I've ever wanted to see in my life, so that's pretty important. Adaptation to environmental stresses like drought. The first year that Jared and I farmed together, we unfortunately had one of the biggest droughts known to Oklahoma history in 2012, and so that was really hard on us as first-time farmers, but definitely hard on our pocketbooks as well. And desirable nutritional characteristics. So you can plant those plants and have the nutritional aspect that marketer or that buyer wants to have. What are some concerns? Some big ones that we've seen and that have had studies on them might be an impact on human health. It can then be resistance to pests, which we'll talk about. And then there are some ethical and religious issues that go along with GMOs as well. So marker-based selection, what are those? It's just a molecular marker that DNA segments that then facilitates the selection of those useful traits within that breeding process. It's a little bit faster and more consistent when you're looking at the selection part, and it's not limited to the transgenic plants as well. So there's a lot of different ways to look at that. And when you look and read in your book, you'll understand a little bit more of those marker-based selections and why we use them, and how important they are to plant production. So, what's the difference between what are some traditional breeding aspects versus genetic engineering aspects? Traditional, we're looking at a couple of years to release, maybe 2 to 5 years. Has many genes. Okay, so again, when you look at this next graph, I'm going to show you if you're breeding for one gene, you're going to get that one gene, but then it's going to have more genes with it. It will use only the genes of the related species, and it's not prohibited in the United States. Okay, so you can use traditional breeding within organic agriculture. Genetic engineering, however, there's a faster release of varieties, so we can breed them and get them to producers pretty quick. Those transfers of genes are very, very few genes that we want. So it's not having to deal with the many genes that then are put into that plant, and it can be used for genes of any species of plant. But unfortunately, it is then that kind of entails it to be prohibited within organic agriculture. So people within organic agriculture cannot use genetically modified organisms or engineering. So here's that graph that I was telling you about. So if you look at the top there, the traditional breeding, so we're looking at the donor, the recipient, and then the new variety. So here's my donor on the left, far left top. If my desired gene is that green spot, I'm going to cross it with the recipient. I'm then going to get that desired gene, but then I'm also going to get a lot more genes with it that maybe I want, maybe I don't want. But that genetically engineered gene on the bottom left, you're going to see that green gene again, that desired gene. It's going to be isolated, like we talked about, into the recipient, and then I'm going to get that desired gene and nothing else. Okay, it's one gene transfer. That's it. I'm going to have the same exact gene that I wanted with the variety that with that donor, those donor genes that I wanted as well. So, how is marketing all intertwined with all of these modern crop varieties? Really high value in terms of crop production by the breeders, by really big private, large and private companies. So we'll talk about the big merger that we've had with Bayer and Monsanto and how that's kind of affected seed prices. Again, this week we're going to look at commodity crops and how they've produced with those breeders at universities, which is really important, which is what we're trying to do with a lot of the research that we're starting, again, very, very early. I probably won't be alive when we've actually done this at NEO, when we've actually gotten there, because it takes a long time for that. But and then patenting of those seeds for sale, and then we're really going to talk about how farmers are not allowed to save those patent seeds for replanting or sale. So if I buy a seed in spring of if I buy a seed that is supposed to be planted spring of 2017, and that's what I tell the USDA, I tell the crop insurance, I tell my seed salesman, I cannot then save it for spring of 2018. That is actually illegal, and we'll talk about that in class this week.